When you look at this screen, you see the letter E. You know this because light from the screen enters your eye and projects an image onto the retina. This remarkable layer of tissue processes the image and sends signals to the brain, allowing you to perceive the letter E. When light enters the eyeball, it passes through the cornea and lens, which focus the image on the retina. Retina is the innermost layer of the eyeball and contains the complex neural circuits needed to process visual information. The outermost boundary of the retina contains photoreceptors. Photoreceptors are neurons that have the ability to convert light into electrical signals. There are two types of photoreceptors, rods and cones, named after the shape of their outer segments. Rods have elongated, cylindrical outer segments and enable night vision, while cones have shorter, cone-shaped outer segments and are responsible for color vision. The outer segments of these photoreceptors are crucial for transforming light into electrical signals through a process known as phototransduction. This is because the outer segments contain membranous discs that house a large number of photopigments. These photopigments consist of two parts, a glycoprotein called opsin and a light-absorbing molecule called retinal. When light enters the eye, the retinal absorbs a photon of light and undergoes a conformational change from 11 cis retinal to all trans retinal. This structural change activates the opsin protein, which in turn activates an intracellular messenger protein called transducin. Transducin consists of three subunits. When activated, the alpha subunit of transducin dissociates from its other subunits and in turn activates another protein called phosphodiesterase or PDE. PDE is an enzyme that breaks down cyclic guanosine monophosphate or cGMP into GMP molecules. A high concentration of cGMP molecules is required to keep ion channels in the photoreceptor membrane open. When these channels are open, sodium and calcium ions flow into the cell. This influx of positive ions allows the photoreceptor to remain in a depolarized state with a membrane potential of -30 millivolts, which trigger the release of glutamate at the synaptic terminals. However, when the concentration of cGMP drops due to the action of PDE, cGMP-gated ion channels close, stopping the flow of sodium and calcium into the cell. When these positive ions cannot enter the cell, the membrane potential drops to about -70 millivolts. This occurs because other channels such as potassium selective channels are always open, allowing potassium ions to flow out of the cell. This outward movement of potassium combined with the halt of sodium and calcium influx makes the cell interior even more negative than before. This is known as hyperpolarization. When photoreceptors hyperpolarize, they reduce the amount of glutamate released. Glutamate is a neurotransmitter, which are molecules released by neurons to communicate information to neighboring cells. Once released, they bind to specific receptors on nearby cells, temporarily altering their electrical properties. The release of these molecules depend on the cell's membrane potential. When photoreceptors are depolarized, glutamate is released into the extracellular space. However, when the photoreceptors are hyperpolarized, there is minimal glutamate release. The glutamate released by photoreceptors bind to two types of bipolar cells, on bipolar cells and off bipolar cells. On and off bipolar cells respond differently to glutamate release because they contain two distinct types of glutamate receptors. Off bipolar cells have ionotropic glutamate receptors, which are excitatory in nature. When glutamate is released, it binds to these receptors, causing the opening of ion channels and an influx of positive ions. This depolarizes the off bipolar cells, making them more active in the dark. In contrast, on bipolar cells have metabotropic glutamate receptors, which are inhibitory in nature. These receptors do not function as ion channels. Instead, when glutamate binds to them, they activate a messenger molecule that closes cation channels. This inhibition is relieved only under bright conditions when glutamate release decreases. The opening of these cation channels cause the on bipolar cells to depolarize, making them more active in the light. To summarize, Off bipolar cells depolarize in response to glutamate and are more active in the dark, whereas on bipolar cells depolarize when glutamate release decreases and are more active in the light. When bipolar cells are depolarized, they trigger a response in downstream retinal ganglion cells. Retinal ganglion cells are neurons that transmit visual signals to the brain. On bipolar cells activate on ganglion cells, while off bipolar cells activate off ganglion cells. These two circuits allow the brain to distinguish between light and dark stimuli coming from different regions of the visual field.